In the midnight ocean, a tiny male follows a chemical trail toward a female glowing like a distant lantern. His journey ends not with courtship, but with teeth fastening permanently into living flesh. If you saw the pair drifting together, they might resemble one animal carrying a strange scar. Yet the scar has eyes, gills, and a reproductive system built for an astonishing biological bargain. This is the central mystery of some deep-sea anglerfishes. Mating can erase the boundary between two bodies. Only certain ceratioid anglerfish lineages practice permanent sexual parasitism, while related species mate without lifelong fusion. Understanding the extreme form begins with the enormous, dark habitat where potential partners are widely scattered. Here, finding food is difficult, but finding another compatible adult may be even less predictable. A male that releases a female after contact might never encounter another before his limited reserves fail. A female that loses a rare visitor may produce eggs without receiving sperm when conditions finally favor spawning. Natural selection, therefore, worked upon repeated reproductive failures, not upon any conscious decision to merge. Across generations, traits preserving a successful encounter could spread when they produced more surviving descendants. The resulting bond looks monstrous through human eyes, but evolution measures continuity rather than appearances. Our investigation follows that bond from first detection through attachment, fusion, and its reproductive consequences. Ceratioid females are generally larger than males and carry the fishing apparatus that gives anglerfishes their name. Their modified dorsal spine supports a lure, often illuminated by symbiotic bacteria in several well-studied groups. Males of many species mature at small sizes with enlarged sensory organs and reduced feeding structures. This difference reflects specialized reproductive roles. Although exact anatomy varies substantially among families and species, some males possess large nostrils suited to detecting dissolved chemicals released into moving seawater. Others also have well-developed eyes, which may help recognize silhouettes, light, or species-specific lure patterns. Direct observations of initial encounters remain scarce because these animals occupy deep, expansive, difficult waters. Researchers instead combine preserved pairs, anatomical sections, genetic evidence, and occasional deep-sea video. Those sources establish permanent attachment in several species, but they do not reveal every step of courtship. A searching male must locate the correct species before currents erase or confuse the available cues. Imagine you entering a dark landscape where every direction moves and the nearest partner could be kilometers away. The male's sensory investment turns faint information into action, guiding swimming toward a promising chemical gradient. Upon contact, he tests the female with sensitive structures before biting, reducing the cost of a fatal mistake. His jaws secure him against currents that would otherwise separate the pair within moments. In temporarily attaching species, the male later departs, after transferring sperm, preserving an independent existence. In permanently attaching species, tissues at the bite site begin an integration unmatched among vertebrate mates. Skin layers join, blood vessels connect, and the male eventually receives nutrients through the female's circulation. His digestive system and several organs regress as independent feeding and movement become unnecessary. Testes remain functional, transforming the attached male into a dependable source of sperm. This sequence links cue, action, adaptation, and consequence with unusual clarity across the reproductive cycle. The female gains reproductive assurance, while the male gains access to a partner he might never replace. Neither benefit makes the process painless or purposeful. It describes differential survival and reproduction over generations. The verified pattern is remarkable enough without claiming that every anglerfish male becomes a permanent appendage. The deepest puzzle is how two vertebrate immune systems tolerate tissue fusion without rejecting the foreign body. Ordinary vertebrate transplants provoke immune recognition because cells display molecular markers identifying self and non-self. Permanent anglerfish pairs somehow cross that defensive barrier while maintaining enough protection against infectious threats. Genetic analyses reveal major alterations in immune-related genes among lineages with obligate permanent attachment. Some lack functional components associated with antibody responses or the destructive activity of certain immune cells. Species with temporary attachment retain more conventional immune machinery, creating a powerful evolutionary comparison. This correlation supports immune remodeling as an enabling adaptation, but it does not reconstruct every historical mutation. Scientists cannot watch millions of years of change, so relationships among living species provide indirect evolutionary evidence. The pattern suggests that increasingly intimate attachment evolved alongside reductions in pathways that would attack fused tissue. Whether immune changes preceded fusion or intensified after attachment began remains an active question. The ocean may reduce exposure to some pathogens, yet deep water is not biologically sterile. Anglerfishes, therefore, must balance tolerance of a mate against defense from microbes, parasites, and damaged cells. That balance probably involves immune functions more nuanced than a simple claim that immunity disappeared. Preserved specimens show different degrees of connection, from fresh bites to fully integrated vascular unions. These snapshots suggest a progression, although they rarely establish the exact time required for fusion. Histological sections can reveal joint tissue and blood vessels, but preservation may distort delicate boundaries. Genetic identification matters because severe sexual dimorphism once caused males and females to be classified separately. Matching attached males to females corrected those errors and clarified which species use permanent unions. Even now, sparse collections can make a rare behavior seem absent from a species where it simply escaped sampling. Conversely, one attached specimen cannot establish how frequently attachment succeeds across an entire population. Multiple males sometimes attach to a single female in species capable of supporting several partners. That arrangement can increase available sperm, while each additional male also draws resources from the shared bloodstream. No evidence shows the female directing attached males individually. Circulating hormones may coordinate reproductive readiness. When her ovaries mature, endocrine signals reaching fused males can stimulate sperm production at the necessary time. The pair then releases gametes into open water, where fertilization occurs outside their bodies. Eggs often enter a buoyant gelatinous sheet or veil, later breaking apart as development proceeds. The permanent bond, therefore, serves a brief spawning event that may occur only when energy reserves permit. Extreme mating is inseparable from the ecology of scarcity surrounding both bodies. Food arrives unpredictably as fishes, crustaceans, or other prey pass within striking distance of the female's lure. When you live where meals and mates are rare, holding a successful encounter can outweigh extraordinary anatomical costs. The female's lure triggers prey approach. Her rapid mouth expansion captures food, and stored energy supports egg production. The male follows reproductive cues, bites, fuses, and converts his remaining metabolism into reliable fertilization. Each behavior answers a different scarcity, linking feeding opportunity to reproductive timing in the same drifting habitat. Sexual size difference also reduces competition because the diminutive male requires far less food than another large hunter. His small body can mature with limited resources, travel efficiently, and depend on the female after attachment. This specialization becomes dangerous if he fails to find a mate before exhausting his reserves. Not all males are incapable of feeding, and researchers avoid applying one species condition to the whole group. Likewise, not every female carries an attached male when captured, which may reflect season, age, or failed encounters. The evolutionary outcome is therefore a mosaic rather than a single ladder toward greater dependence. Independent mating persists where encounters are frequent enough, or where ecological costs make permanent fusion less advantageous. Temporary attachment persists where secure sperm transfer helps, without requiring complete vascular integration. Permanent fusion appears where reproductive assurance repeatedly outweighed immune remodeling and lifelong physiological dependency. These alternatives turn ceratioids into a natural comparison of solutions to the same deep-sea problem. They also show how selection can modify immune systems when reproduction depends upon accepting genetically different tissue. Medical researchers study such mechanisms cautiously because distant evolutionary solutions rarely transfer directly to human treatment. Life can preserve individuality, suspend it temporarily, or build reproduction around a lasting physiological partnership. Future evidence must begin with more encounters observed from approach through attachment in undisturbed deep water. Long-duration submersible video could test which cues guide males and how females respond before the bite. Fresh specimens examined across attachment stages could clarify tissue changes without relying entirely on preserved snapshots. Comparative genetic work across more species could refine when immune alterations appeared on the Ceratioid family tree. Researchers also need population estimates revealing how often adults meet and how frequently attached pairs reproduce. Those measurements would test whether mate scarcity alone predicts fusion or whether other pressures shaped its evolution. They might reveal environmental thresholds separating independent males, temporary partners, and permanent sexual parasites. Climate-driven changes in oxygen, temperature, and food supply could alter those thresholds, although present effects remain uncertain. Careful language will remain essential because dramatic anatomy easily invites stories stronger than the evidence. The male does not knowingly sacrifice himself, and the female does not deliberately consume his identity. If you return to that drifting silhouette, the apparent scar now records a solved encounter and many unresolved mechanisms. Its joined bloodstream proves physiological fusion, while its evolutionary path must still be reconstructed from comparative evidence. The glowing lure illuminates prey for moments, but the surrounding darkness conceals most meetings from human observation. Somewhere beyond the camera's reach, males may be searching, rejecting mismatched females, attaching, or dying alone. Every successful union preserves two genetic lineages while producing a body arrangement unlike either ancestor alone. Its extremity emerged because repeated scarcity rewarded permanence, sensory precision, and tolerance at the boundary of self. The scientifically honest ending is stark. Permanent fusion solves mate loss for some anglerfishes, but its full origin remains unseen.